All right, good morning, guys. I just completed a tutorial, live Zoom, for people on this activity 7.2.1, black plus black equals red, which is the dihybrid Punnett square. Dun, dun, dun. I wanted to give you guys a quick tutorial video if you so chose to watch it and wanted to help you with completing the dihybrid Punnett square. If you didn't get to watch the Zoom and you want to see how it's done. These can sometimes be complicated. I know I told you I'd give you the answer key, but I want to share with you how to do it. So here's your chance. All right, so I have this up on a Google Doc so that I can work with it. And I want to scroll down to the basic information here first. Reviewing the genetic codes for coat color and polled or horned and Angus cattle, because this is the example that we're using today. So the coat color genes are black, uppercase B, red, lowercase b, and black is dominant to red. And then the horned or polled gene, uppercase P is polled, lowercase p is horned. And I tell you, I just had done this, walked through people with this, and in Google Docs, there's a feature where you can undo changes and redo changes. So I fixed that lowercase b is horned, and then when I walked through it, that's why I just fixed it on the sheet. I couldn't handle that. So polled, uppercase P is dominant to horned. But we're just going to walk through this and redo all the changes that I did with these fine folks that were on the Zoom, and that way less for me to do when I'm going through it with you. And I can go through it fast. Yay! Okay. Screencasts only have a 15-minute time limit, by the way. So, well, the free version. So stop wasting time, Mrs. Burnson. Okay, so I'm color coding these for you so we can do it. Now, the great thing about this is that the scenario tells you have two heterozygous animals. So that means we only have to identify the potential genotypes once, and we use it for both animals. And the other thing to note is that the genotypes that are here. So when we do this, we use what's called I use the FOIL method, used for distribution in math. First, outside, inside, last. And it helps us to get each potential combination when we're using it with this here, so that we have all the combinations possible of a B and a P, and make sure we don't miss anything. So there's 2 letters of each, which means there's 4 possible combinations of B and P. So I'm color coding, and I'm color coding the B. First B, the uppercase B is red, the second B is teal, that's lowercase, the first P, uppercase, is yellow, and the second P, lowercase, is purple. So to get the first one, we are taking the first letter from each of the genes, which is uppercase B, uppercase P. Then we have the outside letters, which is uppercase B and lowercase p. Then we have the inside letters, which is lowercase b, uppercase p. And then we have the last letters, which is lowercase b and lowercase p. Great, right? That okay? So first, outside, inside, last. That ensures we don't miss any possible combinations. That's very important. So again, this time, you guys get a freebie, and it's quarantine time, so we got to take it easy on you. We are putting the same letter combinations down the side because our 2 animals happen to have the same genotype, the parent animals. So that's a bonus. So now when we're filling these in, we're going to fill them in the same way we'd fill in a monohybrid cross. That anything that is above a column, those letters get transferred down into the columns. Anything that is to the left of a row gets transferred across that row. And when you put the letters in the boxes, B's have to stay together, P's have to stay together. B should always be first because we want it to look similar in order of the parent genotype, so never transpose the order. You also want to make sure that uppercase letters stay before lowercase letters within the B's and the P's. So I'm going to fill them in here for you. And you can see my work coming across. Okay, so now we scroll down and it tells us that it wants to know the frequency of each of the genotypes. And these are all the possible combinations on the left over here that you could get from the table. Sometimes it's confusing to make sure that you have all of them or you've gotten all your combinations. So I am going to continue to color code here. The color codes in the table and down below do not have anything to do with the color codes that we initially used here. And please do not feel like you have to color code. You don't have to color code anything when you're turning in your assignment. So the other thing that you should know when you're putting the frequency in is that your total is going to be 16. So you can add up your numbers. If you get 16 numbers, then you know that you've done this correctly. So my first one is uppercase B, homozygous for both traits. And there is only one individual that has that. Second one, homozygous dominant for B and heterozygous for P. There are 2 individuals that have that. Third one, homozygous dominant for B, homozygous recessive for P. We have 1 individual with that trait. Or 1 potential offspring, we'll say, because this is probability, not actual offspring. Fourth one, heterozygous for B, homozygous dominant for P. We have 2. And you can see a pattern here, folks, the way it's going across the table. A lot of times it does pattern like that. Not always the same pattern, but look for patterns in your tables. That will help you as well. Next one is homozygous for both traits. It's a popular genotype. There is 4 of them in this table as a possibilities for offspring, which makes sense because the parents had that genotype. Next one is homozygous for B and heterozygous for B, homozygous recessive for P. There is 2. Next one, homozygous recessive for B, homozygous dominant for P. There is 1. Next one, homozygous recessive for B, heterozygous for P. There is 2. And lastly, we have homozygous recessive for both traits. There is 1. Now we want to go through and we want to look at what is the phenotype, or what each of these genotypes will show or express in the offspring and what it looks like. So you can either go across with the traits and say, okay, the homozygous dominant B is black and the homozygous dominant P means polled. Or you could go down and do all the ones that are black versus red and then polled versus horned. That's what we did today. We went down, and so we'll go through these. We did make a mistake, and we had to go back and correct our mistake, which sometimes happens. You got to double check your work and keep working your way through the different components. So first one is a black. Second one, homozygous dominant black. Third, homozygous dominant black. Heterozygous is also going to be black, black, and black. And then we have 3 at the bottom that are red because they're homozygous recessive. And when we go back through, the first one, homozygous dominant, is polled. Heterozygous is polled. Then we have a homozygous recessive that is horned. This is where I made my mistake. This should say polled, but it said horned. We'll go back and fix that in a minute. There we go. We made a mistake again. That one is polled and it said horned. We switched those last 2. And then we're coming down here to the next part of the table. But as I said, we made a mistake and we came back and fixed our mistake. So now we have the right words here. So we've got the last one is horned, and this one up here is horned. And there should be one more we did make a mistake on too. We'll go back and fix that in a minute. So down in the second part of the table, it wants to know the probability. So we count out the number that have each potential phenotype. And again, we're using color. Again, not associated with the colors in the table. But I highlighted all the ones that are black and polled and added up the numbers from the frequency. And there's 9. So 9 out of 16. And then it asks the probability as a percentage, and that would be 56.25. Next, we went for black and horned. There is 3 out of 16. And again, 3 divided by 16, multiplied by 100, that's 18.75. Next one, red and polled. 3 divided by 16 also. So we have the same percentage, 18.75. Then lastly, 1 out of 16 is red and horned. That percentage would be 6.25. So the last 2 questions, what is the probability of the farmer's bull and cow having a horned red calf? 6.25%, or 1 out of 16. And then it asks, what are 2 changes the farmer should make to ensure he does not produce a horned red calf again? We came up with a couple of suggestions. Don't keep any horned animals, or purchase a homozygous polled and black bull or cow. I should say black and homozygous black and polled bull or cow. And that matched up with what the answer key suggested as well. So folks, we've just gone through dihybrid Punnett square. Please reach out if you have any more questions. This went pretty well, and hopefully you get the hang of this a little bit and understand how it works. As I told the people on the Zoom, you will not have this on any sort of quiz during the quarantine. Occasionally I'll give it on a quiz during an in-class type of class, but I know this is challenging. This is kind of the upper level of some of the things you learn in high school about genetics, and it can be complicated, so I don't want to overwhelm you. I love teaching about genetics. I think this stuff is cool, so please hit me up if you have any questions or other things that are wandering into your mind about genetics as you're working through some of this stuff this week. All right, have a great day. Bye.